It is my honor and pleasure to welcome our guest and the NIPTE community to this 2020 NIPTE conference. I wish we can see more clearly now. Indeed, let's hope our country allocates significant resources to addressing our supply chain challenges and help prepare for the next public health emergency. We still have significant challenges remaining in this public health emergency. With Professor Muzio as the new chair-elect of NIPTE, I can't but help reflect back those days while I was at FDA, and I had invited him to come and spend some time with me to help think through a challenge that we were facing of dissolution testing. So this is a reflection on that time, and I look forward sharing some insights about the next NIPTE spiral as I depart NIPTE. These are my departing thoughts. Over the past decades, I have learned a few lessons while working to overcome the systemic, harmful, societal underappreciation of the role pharmaceutics, industrial pharmacy, and pharmaceutical engineering plays in public health. The little secret is a flaw in the never let a serious crisis go to waste strategy. As we struggle to see more clearly the ways to boost our innate immunity, we must overcome the systemic immunity to change. That's the theme of my talk, and I'll elaborate on this theme to explain what I mean. Perhaps most of you may not know that the formal launch of FDA's Process Analytical Technology Initiative was scheduled to occur on September 11, 2011. We assembled in a hotel for our workshop. We had guests from Pfizer and other companies participating in that workshop. After the initial presentations by Dr. Woodcock and a few others, at the coffee break, we learned that the world has changed. Obviously, the workshop was ended at that time. But our commitment to progress on that was just strengthened. 16th of November 2001 was the FDA Science Board meeting, and we went ahead with that. In October 2001, the US Command and Control held a workshop or symposium on sense making. That report contained this model of sense making. 3 domains: cognitive, information, and physical. We begin in, where do we begin in this cycle? Let's begin in the cycle. Once we have taken some action, we have objects and events. Those objects and events generate data which is representing of those objects and events, and when we place the data in context, we generate information that informs our awareness. And our awareness is influenced by our emotions, beliefs, and perceptions. If our awareness or consciousness is narrow, we have a very narrow awareness. If our consciousness or awareness is broad, we have broad awareness of the situation. Our situation awareness has to be shared within a community. It interacts with our prior knowledge. I have inserted new prior knowledge because we have written a paper on that in the context of generic drugs. It interacts with our prior knowledge and mental models to generate some understanding. Our understanding generates the basis for making meaning or sense making. And these aspects also have to be shared within a community for them to build consensus. From that, we arrive on a judgment and intent command. The intent command then is transferred into the information domain with our planning directives, and from information domain through synchronization to action, it comes in the real world. Sense making is a very important topic, especially in high complexity, uncertainty, and chaos. Your beliefs are cause maps that you impose on the world after which you see what you have already imposed. Common sense is a collection of prejudices acquired by age 18. Common sense is not so common. At the FDA Science Board meeting in November 2001, we heard that phrase, don't use and don't tell, with respect to innovation and new technology application in pharmaceutical manufacturing. That sent a shockwave through the system. I have collected a slide from a USP workshop on continuous manufacturing that Professor Muzio and myself participated. And my thoughts were, we need to be thinking about taking pharmaceutical continuous manufacturing from extraordinary to normal, easy, and rewarding. And I recalled this paper I wrote before in 2005. The nation needs a comprehensive pharmaceutical engineering education system. This was in part to support the justification for CSOP's establishment of an engineering research center on pharmaceutical engineering. So that was 2005. And since then, I've been on a journey to think about behavioral economics and management of pharmaceutical QBD in my private practice, and the caution is beware of regulatory defaults. What are regulatory defaults? Defaults are the decision criteria that we came up through a consensus opinions of 10x scale-up factor of 10x, 3-batch validation, and so forth. These are the defaults. These we have to be very careful about. At the April 9, 2002 Science Board meeting, FDA Science Board meeting, Janet Woodcock herself reminded the community that the major barriers to adoption of new technology was industry concern about regulatory implication of results. Closer scrutiny will reveal variation in existing products missed by sampling, and there will be delay in approval of new products. With the emerging technology team and so forth, we have addressed some of those challenges, especially with respect to delay in approval of new products. But you have major legacy products where closer scrutiny will reveal variation in existing product missed by the current quality control testing, which is essentially compendial testing or market standard testings, which are the minimal standards. They were, I posit, never intended to be QC test batch release, and there is a disclaimer to that effect in the USP. So Dr. Woodcock illustrated that with an example that about 4% of batches fail, although there is no difference than from passing batches. This is something you will have to address as we go along. In 2005, I moved to Sandoz Novartis to help them secure their biosimilar portfolio, establish that quality by design platform for biosimilar development, and push for establishment of a regulatory pathway from the industry side. I was recruited then by Philip Morris International to work on two interesting projects. One was to help establish a platform for making vaccines in tobacco plant, and we focused on making vaccine for pandemic H5N1, and that system is now focused on COVID vaccine, and also worked on modified risk tobacco products. I had not anticipated returning back to pharma, but I did. Reconnected with India, and my practice was focused on India. And I realized the challenges of assuring quality and assuring assurance of data integrity, and established my own consulting firm to help many companies do that. So in 2015, I thought it would be useful to start capturing what I learned and share it with the communities that I'm a member of, and I did that on LinkedIn. And my journey was called Schrodinger's Cat, and my journey from 2015 to 2020, and it essentially ended in December of 2019. So in 19 years since FDA PAT initiative, what have I learned is what I want to share with you. Vision 2020, I can see clearly now, was the vision statement I created for FDA's PAT subcommittee. And the bullets were quality and performance by design, continuous real-time monitoring of quality specifications based on mechanistic understanding of how formulation and process factors impact product performance, high efficiency and capacity utilization, real-time review and inspection from Rockville, White Oak, New Jersey, wherever FDA is. And today, we are in the mode of virtual inspections, and we are still working on all other objectives listed here. Manufacturing is a stepchild of the sector. Much of the attention goes on marketing and R&D aspects, but manufacturing gets attention when there is an FDA warning letter. FDA Voice, January 2017, a review of CDER's novel drug approvals identified that the major problems for complete response letters were manufacturing deficiencies. And one major company board of directors asked me to do an after action review because they had acquired a smaller company and ran into multiple problems. Why multiple complete response letters? FDA inspection observation of an acquisition by a big pharma. What did we find? Quality systems were immature, complex, highly paper-based. QC lab systems were not compliant, and lab extremely cramped and inefficient. Lack of process development change history, immature business processes, staff capabilities were lacking, facilities and equipment not well designed for commercial manufacture, contents of US filing not aligned with big pharma standards, processes, specification limits, process description controls were all a mess. But this company paid $3 billion to acquire this after extensive due diligence. What did they miss? How did their system miss? And that was the reason for this after action review. In 2016, I came across an FDA presentation, which was very surprising to find FDA listing 6 companies which they think had achieved quote unquote 6 Sigma level of quality. Not the 6 Sigma methodology, but the low error rate of 3.4 defects per million opportunities. And one of those companies listed was Amgen. So I was intrigued. I don't have any association with Amgen, but Amgen was deeply involved with our efforts at PAT. For example, James Seeley had come and made a presentation at our Arden House conference on their approach to process characterization and failure mode effect analysis, in essence, quality by design principles that they have been using. So, taking available information from the public domain, I did an outside-in look at why did FDA list Amgen as one of the companies which has achieved the low error rate of 6 Sigma or 3.4 defects per million opportunities. I will not elaborate in the details of the approach Amgen used. I have written a paper on this, how to break the 2-3 Sigma barrier. If you Google that, you'll get that paper and so forth. So, but I'm just going to highlight the big picture of the Amgen's journey to 3.4 defects per million opportunity. The journey starts with the little secret. The pharmaceutical industry has a little secret. Even as it invents futuristic new drugs, its manufacturing techniques lag far behind those of potato chips and laundry soap makers. What keeps us from changing? We are victims of our own success. Products have long patent protection. We are isolated from economic cycles. Regulatory systems promoted and encouraged to inspect and test quality. There has not been sufficient pain, creating pressure to force us to change. How do we make sense of FDA approved, validated, those phrases? Many companies take on the mindset that the product is FDA approved, process has been validated, so if I don't look, there is no problem. That changed at Amgen in 2006, and they came up with the realization that we are here to serve patients. We cannot be waiting for FDA inspectors to come and tell us that our corrective and preventive actions are ineffective. We need to investigate and correct and prevent errors before FDA inspection. We should not depend on FDA inspection. So they undertook a journey from 2006, institute a risk-based classification system to focus on high risk, improved trending and non-conformance, improved management review. In 2010, they standardized root cause analysis. 2011, they developed technical writing course for investigators, involved quality science in investigations, and not just do investigations for the sake of doing investigations, but get to the root cause, or taken a multivariate approach to cause and detection and so forth. And in 2012, they initiated investigated mentoring, qualification, and certification program. Amgen's journey took over 10 years to get to 3.4 defects per million opportunities for every process parameter, every deviation possibility, and so forth. So what can we learn from that? Clearly, when you succeed, the power of error reduction is profitable and it is very satisfying. What I would like to propose to you is the mindset. If I don't look, there is no problem. That's a mindset one. To a problem-solving mindset and prevention of error mindset, we can think about these as stages of human development, of professional development, of individuals and of corporations. Remember, FDA just launched a program on assessing maturity of a quality system. Our system is a socio-technical system. We need to think about maturity of our technology, and we need to think about maturity of our people. So that's the reason why my interest in adult human development and professional development. To be evidence-based, I adopted the constructive development theory of adult human development of Professor Robert Keegan at Harvard, where he has 5 orders of consciousness: impulse control, imperialist, socialized mind, self-authoring mind, and self-transforming mind. The socialized mind is the 3rd order of consciousness. 4th order consciousness is self-authored, and prevention of errors is what I call 5th order, is self-transforming mind. So Amgen's example illustrates a self-transformation at Amgen, which required a 5th order of consciousness, at least in a core group of leaders there, and to bring the rest of the organization to a problem-solving stage, which is the 4th order of consciousness, as you saw from the list of action items they took. So this is Professor Keegan's constructive development theory. Constructive development means we develop by making meaning, and the meaning we make as we develop changes. And I think that's the foundational basis of constructive development theory. And immunity to change is also his concept. And he defines immunity to change as a change prevention system, feeling system, and knowing system. And those 3 constitutes immunity to change. We saw the Amgen example, what was holding them from changing. And clearly, feeling system had to be there, is because we are here to serve the patients was the key feeling that they adopted. And knowing system was to know how to solve how to investigate across the organization. So in that sense, one can think about constructive development theory of professional development of individuals and corporations in this manner. I've used the phrase "don't use, don't tell" from Pfizer at FDA Science Board meeting in 2001. And if I don't look, there is no problem, from Amgen's example of being socialized mind. Socialized mind is, tell me, it's FDA approved, it's validated, that's done. But taking ownership of quality, not depending on FDA inspection, is the 4th order of consciousness. You're self-authored. You're self-authoring your own standards above and beyond the USP and FDA. And taking a higher order of consciousness is to prevention, because not everyone will be at the high order of consciousness. The leadership has to be, and they have to guide the organization towards problem solving. And that's what the Amgen example illustrates. Human development, or lack thereof, at times of challenge and controversy. We will not solve the problems tomorrow with the same order of consciousness we are using to create the problems of today. We must be the change. To do so, we must pay attention to a canary which is tweeting in the coals of our mind. We must agree to see the elephant in the dark, removing the blindfolds to begin to see an elephant in the dark. Now I wish to contrast the Amgen example, which is a biotech example, with a small chemical modified release example. In case is metoprolol succinate. Now, what's the difference? Biotech products don't have product monographs in the USP, so they don't have. Plus, they don't have to deal with the solid state properties. Remember, one of the biggest challenge we have in solid dosage forms is that our certificate of analysis for our incoming raw materials is based on identity, purity, and strength, but the physical attributes necessary for processability are not adequately addressed in most cases. So that's our starting condition is variable, and then we have adopted a way to fix process time. We blend for 10 minutes, we granulate for X times, and so forth, because that was the only way to quote unquote validate. And the whole idea of quality by design was to change that, bring real-time measurements so you can blend till it's homogeneous, you granulate to the right particle size, and so forth. That was the whole idea. Now, the situation I want to elaborate here is I was at FDA in 2005, so I was aware of the review practices. There was an outcry, an FDA response to the outcry because of all the recalls, import alerts, and the hoopla that occurred because of product failures. And then I had an opportunity to help a company resolve a complete response letter for this product. And in fact, we used that as a case example for NIPTE training when we did the training at Sun Pharma. So the point here is FDA spokesperson came out and said in 2013, recalls of this product were results of the company's own test. Think about that. What do you mean, company's own test? Christopher Kelly, a spokesman for FDA, acknowledged in a statement that there have been challenges in consistently manufacturing metoprolol succinate and said the agency had consistently monitored manufacturing of the drug to prevent problems. He said in 2013, the FDA undertook a multidisciplinary investigation of the efficacy and quality of each of the versions of metoprolol succinate available in the United States. The agency concluded then that all versions, including those made by company X and Y, were bioequivalent to the brand name version. The reason I'm blanking out is our intention is not to point fingers. Our intention is to understand the underlying cause of the problem. He said that recalls were the result of the company's own tests. If I'm not mistaken, I have to confirm with Jim Pauly. The biostudies for these products were done at University of Maryland. That's what I think, but I'm not sure. This problem is not new. On Joe Gradion, on October 21st, 2007, we wrote to alert FDA and its Office of Generic Drugs about problems patients were reporting with this medication. We were told, in a sense, we'll get back to you. We heard nothing. So that was 2007 situation. But then in 2014, another recall reveals the dark side of generic drugs. Here we go again, another generic recall from an Indian company. The FDA announced problems with metoprolol succinate 25 milligram extended release tablet, and the company is in the process. What's going on with this heart medicine? So if the problem was the company's own dissolution test, this is across many companies' across many companies. So the common cause is the test, the dissolution test. How do we establish the dissolution test? And what is the basis of that dissolution test? FDA always wanted a discriminating dissolution test, when perhaps if you test two lots of the USP calibrator tablet, they won't meet the F2 criteria probably. So that's how variable the calibration tablets is. This is a fundamental problem that still exists, and Professor Muzio had come in to FDA almost 20 years ago, more than 20 years ago, to help think about that problem. And we are still talking about that problem. That problem has not gone away. What we know about dissolution tests, especially in the context of in vitro in vivo correlation, we know that in vitro in vivo correlations are formulation specific. So if you have an in vitro in vivo correlation for formulation 1, it doesn't automatically transfer to formulation 2. And furthermore, I think the idea of dissolution testing for generics is FDA's tendency after 2007 had become that they imposed on the USP criteria, and then they realized that literally every generic modified release needs to have its own monograph. So instead of having one USP monograph for a physical attribute such as dissolution, now we have polygraphs. Um, uh, and if you go back and look at the FDA review occurring in 2005, 6, 7 time frame, they were struggling. The high variation of dissolution data is due to manufacturing process. How do we know that? That's just a statement. When the ER tablet, different number of broken down. However, the in vitro variability is unrelated to performance of the product in vivo, because they had done multiple dissolution bioavailability tests. So why are we struggling with getting a grasp on what is the appropriate level of discrimination? On the new drug side, I always used to say, if you have made 5 batches for your multiple clinical studies that you have done, at the end of phase 3, when the dissolution specifications are set after the clinical trials are done, and FDA would set a specification to fail one good lot because they wanted to have a discriminating test. And the reality then is, if you're going to set a specification to fail one good lot, the company never designed the product for that new specification. And although regulatory affairs department will accept it, and then they will find a way to have the same lot of raw materials, do a day shift, and get the best people, and perhaps pay 5 times to manufacture 3 lots, and you're validated for the rest of your life. And then the manufacturing operations have to manufacture this in the stresses of a real world situation. And how representative was the quote unquote 3 qualification batches for the real world situation, where you have to manufacture day in, day out, night shift, and so forth. So these are fundamental issues which have never gone away. Perhaps not gone away, perhaps got compounded. So if I look at the 2018, it's I think 2017 complete response letter that we used, FDA then is insisting on additional dissolution tests as in-process tests. So you're just compounding that fact, that challenge. After in 2013, FDA spokesperson says that the problem was your own test, and the test is common to essentially the same dissolution test across companies. And now you're asking for one additional test as in process. How will they manufacture the product? Is that manufacturable in a commercial setting? That becomes a challenge, and we have to address these challenges. So let me share a big picture view of the sector. One, the US generic drug market in chaos. Indian upstarts rise because FDA has been approving. Generic pill color change may affect medication adherence. In fact, it does more than that. So if you do not provide assurance of quality to patients, even color and shape change results in significant reduction in adherence. So that's a defeat of the public health objective of cost-effective medications. FDA moves to increase generic competition, and our new prior knowledge paper specifically addressed that. How many of the FDA-approved applications are actually commercialized? Answer is that's a big problem there. So number 3, fed up with drug companies, hospitals decide to start their own. I don't know the number of hospitals now. Civica RX is now really emerging as a major model for generic supply in US. Suffocating silence prevented us from questioning the rot in the system. As an independent academic body, have we ever questioned the rot in the system? No, we are just focused on doing what we want, research dollars only. No, we have to change that mindset. That's the only way you can have a seat at the table. Continuous manufacturing has a strong impact on drug quality. That's a favorite topic for NIPTE. But that favorite topic is in the midst of the real world challenges. You have to find a way to justify that solution that is favorite solution of yours to solve real world problems in this context. Companies focused on raising prices, to improve profit, bad eye warning letters, and then you have the FDA seeking maturity of a quality system. On what basis? Remember, system is a socio-technical system. Technical maturity. So technical maturity means maturity of your measurement systems. How mature are your measurement system for physical attributes, for blend uniformity? We have been struggling with that for 20 plus years, and from outside, when you look at it, does this industry know how to blend powders is the question that comes up. We have to wake up. Pharmaceuticals beyond 2020 will need to overcome legacy challenges. Yes, we are excited about advanced manufacturing in the US with real-time and remote control, but there are corporate and regulatory challenges that we need to encounter. I wrote a paper in 2019 for CPHI called Chaos to Continual Improvement, and I've shared that with many of the faculty members. So if you're not, please request that I can share that with you. But I think the journey ahead is chaos to continual improvement, is what I propose. Our system is a social system of law and order. Never let a crisis go to waste has been the basis of how these laws have come about. At every crisis is an opportunity to enact a new law. That's how we have done it. So we have layers and layers and layers of legacy challenges, and some of the legacy challenges are underneath those layers and difficult to solve. Pure Food and Drug Acts of 1906, Food and Drug and Cosmetic Act of 1938. So let's look at this. I just want to point out, I think the 21st Century Cures Act perhaps is an example of perhaps a proactive law, but that's a different story. By the 1930s, it was widely recognized that the Food and Drugs Act of 1906 was obsolete. By 1937, most of the bitter disagreements as to what should replace it had been resolved, but congressional action was stalled. Then in 1938, elixir of sulfanilamide tragedy that FDA calls sulfanilamide disaster tipped the scale, and Federal Food, Drug and Cosmetic Act was enacted. The act that was enacted was already in place, already written up and waiting for a crisis. So did it address this particular crisis? Perhaps not. So we have to listen to the sound of silence here. And the chemist who formulated that elixir put a bullet through his heart. Most people forgot the chemist who formulated that formulation. The answer to the question, why name elixir of sulfanilamide tragedy sulfanilamide disaster? If you Google that, that's what's on the FDA website. Why call it a sulfanilamide disaster? Sulfanilamide drug in different formulations has served many patients. It was the elixir formulation which was a challenge. And this is the beginning of our initial conditions of the blind spot. FDA does not approve a drug substance. It approves a drug product, but it does not even acknowledge that in its vocabulary, new drug application, abbreviated new drug application, and we call our inactive ingredients. And so these are the blind spots that have kept us not thinking about the problem deeply. FDA investigators describe the new drug development process at this company throwing drugs together, and if they don't explode, they are placed on sale. Dr. Messengel, a physician CEO, my chemist and I deeply regret the fatal result, but there was no error in the manufacture of the product. It met our specifications. We have been supplying a legitimate professional demand, and not once could have foreseen the unlooked for results. I do not feel that there was any responsibility on our part. Pleaded guilty to the charge of misbranding because in pharmacy, elixir by definition is an ethanolic solution. So he pleaded guilty to misbranding and adulteration and a paid fine of $26,100 for violating 1906 Food and Drugs Act. It was the formulation and the naming of the formulation that allowed FDA to penalize. If he had used a word solution, instead of elixir, FDA had no means of penalizing this company. It was the formulation name. And yet, in the vocabulary, we have drugs, approval of drugs, not pharmaceuticals or pharmaceutical products. There are many blind spots. Um, in 2015, I gave a talk at USP, and with the diethylene glycol, the 1937 tragedy. When did we incorporate diethylene glycol as a contaminant in glycerin monograph? It was 2007. 70 years to open our eyes. So there are many examples. Many are calibration standards, eyes open when quality becomes a national security issue. That's a dissolution story. What is gauge R&R? Why needed for testing physical attributes? These are many blind spots that we have to confront. When the music stops, lessons in the generic drug scandal, which ushered in an organization called Pharmaceutical Science, which focused on SUPAC and BCS. And remember, the SUPAC modified release guideline was based on metoprolol tartrate extended release, not metoprolol succinate. Why did we choose metoprolol tartrate as an example for justifying SUPAC changes? Why didn't we use metoprolol succinate where the problems have? How generalizable are those things? 21st Century Initiative was a way to reset, but we haven't progressed significantly. And in 2015, FDA had to reset again and seek one quality voice across its review and inspection function, which is the Office of Pharmaceutical Quality now. A remarkable life-saving drug, sulfanilamide, in a new elixir formulation can kill and destroy lives. Taste of raspberries, taste of death. Emotions we feel, that is experience. Why does the US FDA refer to this tragedy as sulfanilamide disaster was a question on my mind at the beginning of this pandemic 11 months ago. Like a migratory bird, can we not utilize our internal magnetic compass to navigate in our darkness, that is uncertainty? Place our attention on written words and listen to the voices of a mother who lost her child, Joanne. Hear the sound of silence of a chemist who formulated the elixir. Voice of a mother. All that is left to us is the caring for her little grave. The first time I ever had the occasion to call in a doctor for Joanne, and she was given elixir of sulfanilamide. All that is left to us is the caring for her little grave. Even the memory of her is mixed with sorrow, for we can see her little body tossing to and fro, and hear her little voice screaming with pain, and it seems as though it would drive me insane. It is my plea, Mr. President, that you will take steps to prevent such sales of drugs that will take little lives and leave such suffering behind and such a bleak outlook on the future as I have tonight. A grieving mother. Listen to the voice of a mother. Certainly, we have a regulatory opportunity in continuous manufacturing, and I'm picking the slides up as a representative of the challenges. So it's not meant to be all-encompassing, but just something I had handy. So I'm using from the USP workshop in 2017 from Janssen. What are the hot regulatory topics in continuous manufacturing? Bioequivalence, raw material variability, bioequivalence between batch and continuous, dissolution testing. What is my advice to NIPTE as I depart? I reflect back on my days at FDA, reflecting back on a visit to DARPA and thinking about the manufacturing challenges in 2005. collaboration to ensure national security through radical innovation in pharmaceutical development, manufacturing, and regulatory process. Large-scale manufacturing. Can we supply vaccines at millions of doses per month? How do we accelerate? What is the basis for warp speed? How do we accelerate therapeutics in a matter of weeks? That was the discussion. At Philip Morris, I took that on to make vaccines in tobacco plant in a rapid way, and that we established that, as demonstrated that to beat cell-based and so forth before a pandemic H5N1. That team in Medicago in Canada are now progressing very well with the COVID vaccine made in tobacco. There is no limit to large scale up because you just grow it. So that is an option that is coming about. But the key is we don't take action in time. We are too much focused on time discounting. Oh, it'll happen next. We have to mitigate risk of time discounting. And the model that we talked about, DARPA, and what happens in vaccine is pre-qualified validation platforms. Remember, we may not think we may think about product is the process only for biologics. But even for pharmaceuticals, any change you do, you have to establish therapeutic equivalence again. You may do it by dissolution or you may do it by bio, but therapeutic equivalence is the foundation. Defense Production Act for devices works because you have standards. You just have to conform to standards. But Defense Production Act is not as easily applicable for pharmaceuticals because yes, you have to have application. You have to have equivalence demonstration, not just make the tablets. So my request or my advice, parting advice to NIPTE is think about pharmaceutical engineering science of therapeutic equivalence. You can't just focus on engineering. You can't just simply focus on science. You have to bring the pieces together. Pharmaceutical engineering science of therapeutic equivalence is the foundation upon which you can think about pharmaceutical continuous manufacturing pre-qualified and validated platforms that NIPTE can contribute. To do that, NIPTE also need to champion a reset of legacy issues, particularly with respect to out of specification finding in the conventional small molecule world, especially for generics. And I would propose that NIPTE should consider generic fee waiver request for NIPTE PCM pre-qualified platform. That would be a game changer. So think about these as your strategy as you think about. And I think that is what my learning is. I'm reminded of an incident of Pure Food and Drug and Meat Inspection Act of 1906. In December 1905, President Roosevelt presented Congress with his to-do list. On his list of items was the need to clean up the packing plants and protect Americans' food and was often adulterated and injurious to those who consumed it. It was after reading The Jungle that Roosevelt had opportunity to jump on the bandwagon of public outcry. He invited Sinclair, the author of The Jungle, to the White House to discuss the evils that were revealed in the book. It was a fictional book. Now, the public outcry needed is on cost, not on quality, or not on reliability. And I think that is something that is a challenge we have to confront. Let me end with this. I wish to see NIPTE having a place at the table. You need to grab a chair when the music stops. Formulate pharmaceutical science evidence in the real world. Position your solutions from continuous pharmaceuticals to pharmaceutical engineering science for therapeutic equivalence in the context of the real world. Personalize the systemic, harmful, societal underappreciation of pharmaceutics, industrial pharmacy, and pharmaceutical engineering. We are in a dire situation. We are in an experience economy. We are in an emotional state. You have to personalize it to the customer, to the patient, to the public. That is essentially important. Thank you for giving me an opportunity to be with you for so many years. Thank you for this opportunity to share my thoughts. I'm always there with you. So if ever you need me, just I'm a phone call or email away. So I'm off to find a jungle, not in a tropical forest, but in my mind to take some time off to write my first novel, um, thinking about the jungle that might open the door to White House to bring a discussion on pharmaceutics, industrial pharmacy, and pharmaceutical engineering for the nation. Thank you for listening.